Bow your heads, please.
Dear Heavenly Father, thank you for this day. I thank you for Miss Ruiz and her patience to teach us. I pray that we'll have a good class. In Jesus' name.
Amen. Thank you very much. Okay, why don't you get your test out if you don't have it out already. And let's go ahead and go through the first test. Heterotrophs. You should have said something about they eat somebody else. Okay, they don't, you could have even said they don't make their own food. I'd be okay with that. Okay. Did anybody answer with consumers and decomposers? You could have said that. There's several different ways you could have gone with that. Mutation. You should have said something about a copying error. You could have said that mutation shows you an abrupt change from the parents to the offspring. You could have said something like that also. Okay? If you have something that doesn't sound like what I'm saying and you want to know if it's right, ask me. Okay? A theory is a well-tested hypothesis or a well-tested educated guess. Photosynthesis is making food from light. Prokaryotic cells? No membrane-bound organelles. That's right. Species, something about they can have offspring that can have offspring. Okay. Number 2. An organism is classified as a decomposer. Is it an autotroph or a heterotroph?
Heterotroph.
Very good, heterotroph. Number 3, a tiger develops lockjaw and can no longer open its mouth. Which of the 4 life functions will it not be able to perform?
Metabolism.
Metabolism, good. Hopefully everybody said metabolism. If you said it can't eat, take off half. Okay? You were supposed to say metabolism because that's one of the 4. Maybe you shouldn't take off a whole half, but did anybody put eat?
Like consumer. So
I put extract energy.
Extract energy from the environment. That's metabolism.
Consume/eat.
Yeah, that's what I said.
Hang on. Let's see what he wants to do here. Hang on. Take off 2 points. If you just said eat, take off 2 points. Okay? Okay, that's half. They're each worth 4 points. All right? Actually, they're each worth 5 points. So I'm not even taking off the full half. Okay? Okay. Let's see. Number 4. In laboratory studies, organisms from group A produce offspring with subtle differences as compared to the parents. Organisms from group B produce offspring that are identical to the parents, and group C are marked differences as compared to the parents, which are asexually reproduced. And that would be B, because they're identical, which reproduces sexually. A, because there's subtle differences, and which one is experiencing mutation? C. I'm going to give you an example of a real mutation I heard of from my daughter yesterday. Her brother-in-law does farming, and he just is very excited because he just got mule-footed pigs. Now think about what I just said. Pigs have a split hoof, okay? He got mule-footed pigs, which means they have a hoof like a horse. And I said to her, that's got to be a mutation. And she goes, yeah, but they breed for it. It's a breed, mule-footed pigs. And so they've taken that mutation, and because it's an actual change in the DNA, they're able to breed for it. Believe it or not, some of your breeds of dogs come from that. When you have real tiny versions, let's say like those little mini pincers, I think that might be like, you know, when you get those smaller ones, frequently you're taking a mutation and you're benefiting from it. That's what. Do you like seedless fruit? That's a mutation that we benefit from. Because can seedless fruit make new plants? But they sure do taste better than grapes with a bunch of seeds in them, don't they? Yeah. So we can benefit from some of the mutations. But the little mule-footed hog just really got me going. I'm thinking, wow, that's somebody breeding a whole breed out of a mutation. So anyway. Okay, let's see. Now we all have to see what a mule-footed hog looks like, right? Let's see, number 5, a biologist studies 2 organisms from the same family. She then studies 2 organisms from the same genus in that family. In which case do you expect the most similarities between organisms?
Genus.
The genus, very good, because that's where they're more similar. Remember, it's kingdom, phylum, class, order, family, genus, species. So they go more similar as you go along. Number 6, a biologist wishes to study how two remarkably different plants react to the same changes in their surroundings. Should he choose organisms from different phyla or from different orders within kingdom Plantae? Phyla, because he wants the biggest differences he can, so you want them from different phyla. Okay? Number 7, why did the law of spontaneous generation survive for so many years? Faulty experiments, or you could have said something about faulty reading of the experiments because people have had a prejudice, so they misread what was going on. Anything that said something like that would be fine. Okay? Number 8, classify the following organisms using the biological key in the appendix. And so for number 8, he is looking for A, Plantae. 8A is Plantae. Now, from there, you knew that it was, well, I don't know. Did you guys know that it was Anthophyta?
Yes.
You got that far? How many of you got dicotyledon A? How many of you didn't get dicotyledon A? Don't count it off. You don't know enough about plants to answer that question. So just leave it alone. If you got that far, praise the Lord. That's good. All right, B, Animalia, phylum, Chordata, and class Aves. I think you can do a bird. So that shouldn't be a problem, right? Okay. Number 9, an organism is these are my favorites. An organism is a multicellular decomposer with eukaryotic cells. So if it's multicellular, right away we know it's eukaryotic, don't we? And it's a decomposer. So what kingdom is it? Who am I?
Fungi.
Fungi, right, okay. An organism is a multicellular autotroph. Who am I? Plantae, very good. I'm a plant. And an organism is a single prokaryotic cell. Who is it? Monera, very good. If we were using the 3-domain system of classification, in which domain would an organism made of a single eukaryotic cell be classified? Eukarya. Very good, very good. Okay, and what did I just tell you? They're worth 5 points apiece. Now that includes each of the vocabulary words are worth 5 points apiece. Okay? Always, always, always start with 100 and subtract what you miss. Never add up to get your score with me. Because I always err to the side of grace. I give you extra points to, just for taking the test type of thing. So always, whatever it is, subtract what you missed from 100. Okay, and that's how you determine your score? All right? Okay. Any questions? If you are not happy with your grade, you need to study the chapter again, study the study guide again, and take the test again. We're homeschoolers. We can do it till we get it right. Yay! Okay, get your book out. And I am going to get to work. I can see too. Oh, there's words on the page. Okay, it's terrible. We are on Module 2, Kingdom Monera, page 37, please. We are learning about bacteria. Are bacteria eukaryotic or prokaryotic cells? Prokaryotic cells, very good, which means they don't have membrane-bound organelles. And so the general name for them are bacteria. And you hear about bacterial cultures. This is what an actual bacterial culture looks like, okay? So if I were in the lab and I were growing a bacterial culture, this is a petri dish. Okay, they're about this wide. I have some, and I meant to bring one to show you, but I brought so much stuff today I forgot, and I apologize. But it's a little dish. It's about that thick. It's about that big around, okay? And there's a little top that comes off. I'll bring one next week and show you. And what they've got in here is agar, which is, actually, we're going to be learning about agar. It comes from an algae, a seaweed, that gels up, and it's got nutrients in it, and so you put it in there and it's got the gel, and then you culture whatever kind of bacteria. Each one of these are where you already read this, so I'm going to say this to you. Each one of these is where the bacteria has gone through binary fission. It's divided and divided and divided until you actually have a group of that bacteria that has continued to divide because the food is available to it. And that's how you would look at a culture in school. The only other way that I know of that we used cultures was sometimes they will put the culture in a test tube that has a screw top, and you would put the agar or a broth in the test tube, and then you would put your You would use what looks almost like a wire on the end of a handle, a little handle. Think about, you haven't done your dissections yet. In your dissections kits, you have a little handle with a little spike on the end that you use as a probe. Well, what you use when you're doing microbial stuff is you use something with a little handle, and then it's got like a little wire on the end. And really, it's not even spiky. It's just a little wire. And the reason is you can dip that in whatever it is that you're testing, and then you can put it on there, touch it to it, or you can touch it to the broth in your test tube that you're going to do your culture in. And then what you do is you take that. I only know this because I'm actually a cell biology major, and I loved my microbiology teacher and the lab, Mr. Wilt, God bless his soul. And you would take the probe and you would flame it in between samples, because you would put it through, you had a flame, a Bunsen burner with a flame going on your desk at all times, because you would flame anything once it touched the bacteria so that you made sure you killed it before you went on to try something else, because otherwise you would cross-contaminate everything. Does that make sense? Mm-hmm. Okay. So anyway, microlab. I found microbiology extremely interesting. I was an animal dairy science major at Auburn because I wanted to be an equestrian vet, a horse doctor. And so to me, I was just going along as an animal and dairy science major, but when I shifted over to the University of Florida, I didn't like how the degree I was in shifted. And so my micro teacher had made such an impression on me, Mr. Wilt. I just loved him. He was like the first one to pull all the stuff that I couldn't understand why they were torturing me with the information. He applied all of it to real scenarios, and I was like, wow, you rock. He was just so cool. And his tests were a learning experience where actually, if you had all the information, you could figure out the answers on the test. I had 102 average in his class. I loved him. I loved, it was a challenge, but I studied real hard and I could figure it out. And it was like a game. Yeah. Now, I am not a micro major. I want you to understand that. I'm a cell biologist. Micro is studying bacteria. I'm not that into bacteria, but he opened the door to how cool all of that stuff could be. I'm a eukaryotic cell biologist. I studied plant and animal and human cells. You see what I'm saying? So that's what my degree work is actually in. But micro was it just rocked my world. He rocked my world. So anyway, so it can be very, very interesting. This chapter is just so short, and but it can be extremely interesting. So bacteria are everywhere. That's basically what it gets down to. I don't care if you wash your hands. They're everywhere. Okay? And what do we call the bad bacteria that cause diseases? Pathogens. Very good. You're allowed to answer. That's good, because we're going to do this together. What do you say? Okay? Pathogens. And pathos actually means disease. When I worked in cancer research at the vet school, I worked for the Department of Comparative and Experimental Pathology. Pathology is the study of disease. Oh yeah, see, I really have done this science thing, all right? And so it's a sickness. You'll just have to forgive me. And so a pathogen is a disease-causing organism. Now, it doesn't have to be a bacteria. I want you to understand that. It could be a fungi. It could be a virus. Anything that causes diseases would be classified as a pathogen. But he's trying to make the point here that some bacteria are beneficial and some bacteria are not. And so the pathogenic ones, you and I would go, bad, right? Like a staph infection or a strep infection or something like that. Give me an example of where a bacteria could be used to benefit us. Where do we see bacteria benefiting us? Yes?
Well, for example, there is a strand of E. coli in our digestive system that helps to break down food.
Absolutely. We have good for us E. coli that live in our gut. Now, some of you are thinking, really? E. coli? E. coli kills people. If there's E. coli at McDonald's, people die, and that's true because there's different strains of E. coli, and you're going to be reading about that if you haven't already. But there's bacteria in our gut that is good for us bacteria, and E. coli is one that lives in our gut. It's what's called an enteric, which means it lives in our gut. They can actually tell if food's been contaminated with fecal material by whether there's E. coli on it or not. Oh, and if there is, or if there's E. coli in your water supply, that means something has crossed over with the sewer to your water supply and you don't want to drink that water. Do you see what I'm saying? So it can be used in that way. So they can be like markers. There's other things though. Before you even get to the intestines, which is where the E. coli live, there are bacteria in our gut that are extremely important for us because they produce vitamin A, shame on me, vitamin K, A and B vitamins. And so if we wipe out those bacteria by like living on antibiotics for an extended period of time, your body will actually become vitamin deficient because you won't be able to produce the vitamins that those bacteria actually help us to make. And that's called flora. Flora. The bacteria that live in your gut that are good for you are the flora in your gut. And so maybe your mom, if you had to take an antibiotic for some reason, your mom gave you acidophilus milk, or she gave you probiotics, which is really a little pill full of bacteria. I don't know if you realize that. No, but it's that important that they're in your gut. A matter of fact, the doctor, if you've got a healthy doctor, not a drug doctor, okay, will tell you sometimes you need more probiotics. They'll test you and they'll go, you really need to get on a probiotic. You've done something to throw off the flora in your gut. And if you do that, everything else starts to go haywire, because if your gut is not taking in the nutrients like it needs to and producing the vitamins that it needs to, your fuel isn't functioning correctly, and therefore, how good's your car going to drive if you start putting garbage fuel in it? Well, how good's your body going to run if you start having a problem with the fuel? Okay? I mean, I have days that I feel that way, trust me. And so that is important. That flora is important. There's another thing. Do you guys like pizza? Yeah. Cheese! There would be no cheese if we didn't have the bacteria, believe it or not, that make these lovely flavors in our cheese. Now, it's not all bacteria, but some of it is. And so differing bacteria and differing what microbiologists call bugs, which are your microscopic organisms, actually working in harmony with the cow, produce all those lovely tastes of cheese. Didn't you ever wonder about the holes in Swiss cheese? Guess what that is? That's where they're respirating, and they make little holes. Yeah! You knew something was up there, didn't you? Okay, so there are beneficial ones that some of them photosynthesize. That's always a good thing. They're producing oxygen, right? There's some that make cheese. Thus, we can have pizza. I like pizza. Some of them actually recycle minerals, which is very, very important. And so there's a lot of really important things that they do for us. Turn to page 38, please. On page 38, it shows us a typical bacterium. I don't know that there is a typical bacterium. No more than there's a typical person. But what can we say about a typical person? Well, what we can say is that if you're a typical person, you have 2 ears, 2 eyes, a nose, and mouth, possibly some hair on the top of your head, eyebrows probably, 2 arms, 2 legs, no tail. Okay, you get the idea, right? A typical person. So at bacteria, it's even harder to get a typical picture of a bacterium because some bacteria have certain characteristics that other bacteria do not. And so what they've drawn for you here is this is just a picture of some different bacteria to show you just how differently they can look. I mean, just whoa. And so what he's got here on page 38 is a picture of a bacteria for you so that we can go through and identify the different parts. And not only do you need to be able to identify the parts, but you also need to know the functions of those parts. I want you to be very clear on that. So let's run through it. First off, there are a couple things that all cells have. And I'm going to teach you those first because you're going to need to remember those when we get over to eukaryotic cells also. What do we call the genetic material in any living thing? DNA. So all cells have DNA. Now in a bacteria, it's actually a circular chromosome, but it's kind of like if you had a piece of string in a circle and then you dropped it on the floor, it might look like that, right? That's what it is. And so this is the DNA. And you know the purpose of the DNA, or the function of the DNA, is the information in the cell. We good? Mm-hmm. So far, so good. The stuff, the jelly-like substance that the DNA is suspended in, what is that called? Cytoplasm. Now this has a word root that you are going to need to know forever in biology. Cyto means cell. And you're going to see it over and over again. So grab hold of that. Write it down somewhere, imprint it. Tell it to your dog, your little brother, okay? Cyto means cell. The plasma of your blood is actually the liquid portion of your blood. And so cytoplasm is the liquid portion in the cell. It's really a jelly-like fluid, but that's where the word came from. Our cells have cytoplasm. Our cells have DNA. As far as I know, all cells have cytoplasm and DNA. They also have like a little skin around the cell that holds the cytoplasm in. What is that called?
Plasma membrane?
It's the plasma membrane, and I'm going to shorten it to the PM because it's really long to write out, isn't it? The plasma membrane. And our cells also have plasma membranes. Not all cells have cell walls. Plant cells have cell walls. Some fungi have cell walls. Our cells do not have cell walls. We have skeletons instead. Okay? So, but all cells have plasma membranes. And the plasma membrane, you can kind of think of it like skin. It has some very cool jobs, though. Not only does it hold everything into the cell, but it also acts like the watchtower. I don't know if you remember in biblical times when there was a city wall, the people on the watchtower determined who came in and who left. Right? Very important job. The plasma membrane is like the guards on the watchtower. They are not only the wall, but the plasma membrane decide what comes into the cell and what goes out of the cell. And it doesn't let the DNA out of the cell. It's like, no, you can't leave, you know? And if it's working correctly, it won't let poisons into the cell. Do you see what I'm saying? So it's got a very important job. It's not just like skin. It actually discerns, when it's working correctly, what goes out and what comes in. Let me grab my eyeballs here, excuse me. Oh, there's one other thing that even our cells have that this has, and it looks like the pepper in the picture. Now, I would ask you what that is, and some of you would tell me, but you might not pronounce it correctly, and I get real weird about this word because it's not phonetic. These are ribosomes. And it's a long I. Everybody say ribosomes.
Ribosomes.
It is not ribosomes, okay? It's ribosomes. And I know it's not spelled like ribosomes. And I'll be honest with you, I went to school at two Southern schools, Auburn University and the University of Florida. So if they're saying it's Southern and that's why we say ribosomes, I don't know. But that's how it was said at both schools. We had a lot of professors there that were not Southerners, so I'm assuming that's how you say it, is ribosome. I just want you to know that. But I do often wonder, wow, I wonder if that's because I went to a Southern school. Let me tell you a quick story. Went to Auburn, 18 years old, far, far from home, okay? Sitting in one of my ag classes, and it was not the first term, but I'm sitting in one of my ag classes, sitting at the front because I could never see. I've had laser surgery. And Professor Tucker, Dr. Tucker, was talking, and this was a 5-day-a-week class, and like about the 4th day, and I keep writing this word, he kept saying the word protein. Protein. And I am phonetically writing it down because I take notes, and I'm a note taker. I still take notes in church. So I am taking notes, just diligently taking notes, and I'm sounding out this word. I'm sounding out this word. By about the 4th day of class, he says it, and I'm sounding out the word, and I go, oh, protein. Protein. And I said it out loud because I was it was like such a revelation to me. I went, oh, protein. And the whole class stopped and looked at me, and I'm like, oh, because it came out of my mouth, you know, and Dr. Tucker just looked at me and kept going. And I was like, oh, there's a language barrier. You know, at that point, I figured out that really there is a slight language barrier even in the South if you don't speak that, you know what I mean? And so, and if you're Southern, please don't take that as an offense because I'm Southern. But I really didn't get it. It took me that long to figure it out. So I frequently kind of question, wow, I wonder if that's because I went to two Southern schools, the way that sounds. But I'm going to teach you how I was taught, so I'm sorry, you're going to have to go from there. Okay, ribosomes. Ribosomes have a very specific job, and the job is in our cells too. Now, the easy version, 2-word definition, they're protein factories. And you should write that down, because you need to have this memorized forever, but it's for sure you're going to have it for the test. But this is true in our cells too. And let me put it to you this way. If the DNA is a cookbook that gives the instructions on how to make something, the ribosome is the kitchen where it's made. How about that? So that's why it's the protein factory. That's where the information on the DNA is made into whatever it codes for. And guess what? I can't remember if it's module 6 or 7, but it's one of those two that you're going to be intimately familiar with that process, okay, in our cells. You won't have to learn it here. But it's the same thing, and it is a ribosome. These 4 things are in all living cells: the DNA, the ribosomes, the cytoplasm, and the plasma membrane. All living cells have those, even the eukaryotic ones. Now, our DNA is in the nucleus. Therefore, it's in a membrane-bound organelle. That's the only difference. Ribosomes are a non-membrane-bound organelle, and that's why both have them. Okay? All right. Then, let's go on. The next thing is we have, if you're a bacterium, you have the possibility of having another layer outside of the plasma membrane. What is that? The first layer outside the plasma membrane. Cell wall. Somebody give me a job that the cell wall has. Good. Say it out loud. Shape. Very good. It gives the bacteria shape so that it actually allows it. Bacteria is plural, and if I mess that up, just let it go if my English isn't perfect on this, okay? Because I've never seen a singular bacterium. They're always in groups, so you'll have to forgive me. Okay, they help with shape. What's another thing they do? Do you remember? Do you want me to list them? So you got shape here. They regulate water absorption. So regulate water absorption. And I've got holds the content together. I guess that doesn't really help us that much, does it? Okay. And the plasma membrane, I already told you that it regulates what goes in and out of the cell. It senses chemicals to decide what should come in and out, and it's made of phospholipids and protein, which right now is like telling you gobbledygook, you know, what does that mean? So we'll learn about phospholipids and proteins when we study Module 6, and that's the cell biology chapter, which I have so much fun, so you will too. I will try, okay? Okay, and then outside of the cell wall, what if the bacteria, not all bacteria have cell walls, not all bacteria have this next part. What's outside the cell wall if it's got it? Yes? Capsule, yeah, go ahead and say it out loud, guys. That's good. Capsule. Okay, so we've got the capsule. Now, the capsule has two jobs, two possibilities. One is it sticks to stuff. Do you want me to just put sticky? Sticky. The book says that it adheres to things. It's sticky. It sticks to stuff. I'm going to tell you one place that you find encapsulated bacteria. On your teeth. Because they stick to your teeth, and that's why we floss. We'll get back to that. You can't brush them out if, when you brush your teeth, you can't get the part that's between the two teeth, and that's why you're supposed to floss, is to knock off those capsule bacteria, the capsulated bacteria that are sticking, that you can't get just by rinsing. We'll come back to that another time, okay? And so the capsule, there's a second job that the capsule has, that if you're a bacteria, you're like jamming if you've got a capsule, all right? What's the second job? What's the second job? Do you remember? Our bodies have things that fight diseases, right? The capsule actually acts as protection against disease-fighting organisms within our body. So let's say an antibody wants to get it. Here, let me say it the way they said it, because they said it so well. Deters infection-fighting agents. Ta-da! Okay, so it actually protects them from our body's immune system when our body is trying to fight it off. If they've got a capsule, there's a chance that they can survive even if our body's trying to fight it. That's the idea. So the capsule is good if you're a bacteria. It's bad if it's us, right? Okay, then it looks like it's having a really bad hair day, doesn't it? And what do we call those weird-looking little spiky hairs? Okay. You know what? I'm going to write it up high because you're not going to see it otherwise. So I'll put it up here. Fimbria. Let me spell it right. Fimbria. There we go. And I think plural is fimbriae. Algae. Fimbriae. Ugh. Anyway, plural fimbria. How's that? Okay, more than one. I don't want to kill the Latin, and I'm sorry. These guys are not just hairs. What are they used for? Really strange. What are they used for? Go ahead, out loud. Grabbing. They grab things. They grab hold. If the thing wants to grab hold, is that a trip? They get their own little arms to grab hold with. And when they're reproducing, they will actually use them to grab hold. I'm not sure what my next picture is. Hang on. There you go. Check him out. I mean, doesn't he look like he's having a really bad hair day? Doesn't he? Yeah. But that shows them to you. And it's multiple fimbria. Anyway, let's see. Oh, we're only missing one part. And I'm going to put it here. Whee! Whee! And it looks like a flag. This is a singular one, so we're going to make it singular. And that is pronounced flagellum. Very good. Everybody say it, flagellum. Okay, kind of like Magellan, right? It's not flagellum. Ugh. It's, you can tell I've heard that before, can't you? It's flagellum. And if it's plural, if they've got more than one, it's flagella. Think Magellan. You guys know who I'm talking about. And so I don't know that I have, yeah, there you go. This guy has flagella, doesn't he? He's got multiple. This guy has one flagellum. You guys tell me, actually all of these are flagellated bacteria. You tell me, what is the purpose of the flagellum?
Movement.
Good. So movement, I like that one because I can spell movement. The book would tell you locomotion. Okay, and that's just a longer word. I think I could spell that one too. But anyway, locomotion or movement. Okay, so that's its job. Now, I just want to throw you out here. When next week, please, Lord Jesus, if we have a sample with somebody that has flagellated bacteria, when you look under the microscope and you see them go cruising by, you won't be able to see the flagellum, but you'll know they're flagellated because otherwise they couldn't swim against the grain. Does that make sense? They'll be swimming against the current, or they'll just go woo, going across the field of vision, and you'll go woo, you know. And so you'll know you see flagellated ones, but usually this is much higher magnification than what we'll be able to do. We go to 400 power. Bacteria are extremely small, and so we won't see that. But when we get to the next chapter and we do eukaryotic cells, under 400 power, you might have the privilege of seeing a euglena, which is a flagellated eukaryotic cell. And the trick is this: they don't wag their tail and swim, which is what I always figured they did, was move their tail and it motors them through. They whip their tail like a propeller, only then the bacterial goes in the opposite direction because of the motion of the flagellum. The bacterial cell rotates because of that movement, and the whole cell propels it through the water. Is that a trip? It is really very, very high tech. And they've been studying, scientists have been studying the bacterial flagellum for over 20 years because it's so small and it's very hard to find out information about it. And it is extremely well designed with over 200 moving parts. It's like an outboard motor. And the flagellum is really where, from what I understand, that's where the intelligent design movement started. As scientists studied this for 20 years and saw that the smallest little organism has this little propeller thing going on that is so technically made and that it wouldn't work if any one of the parts wasn't there, they started to say, how could that just happen? How could it evolve because you'd need all the parts of the motor or the motor doesn't work? See where the intelligent design movement came from? I know a lot of people think the intelligent design movement came from people and studying other things. Came here. Okay, this is it. People started looking at this and going, what they call it is irreducible complexity. Think about what I just said. Irreducible complexity. You take one piece out and the whole thing doesn't work. You reduce it at all, and you reduce the complexity at all, it doesn't work anymore. Does that make sense? Irreducible complexity. That's where the intelligent design movement started. And I want you to be very clear, the intelligent design movement is not a creationist movement. It's an honest scientist movement that looks at this and goes, how could that happen through Darwinian random chance processes? We got to come up with something better. That's all they're saying. Okay, honest scientists, that's it. All right, so that's the flagellum. Okay, so that's the parts. Now, the next thing you have on the top of page 39 is you have the shapes that bacteria come in. And you have to memorize those three shapes. So, the little round ones that look like a little ball, they look like little spheres, literally, are called cocci is plural. Coccus would be singular, cocci is plural. The way I remember that is a circle starts with C. Cocci. Okay? The ones that look like little rods, you tell me, what are they? Bacilli. And I know that actually in Latin means rod, but I don't speak Latin naturally. I'm not a little Roman kid. And so this I remember, they are bat-shaped, aren't they? And that's the beginning of the word bat. Okay? Circle, cocci, bat, shaped bacilli. And then the last one looks like a spiral, doesn't it? And they're called spirilla. Let's make sure I'm spelling it. Plural would be spirilla. Spirillum is singular. Like I said, I've never seen a singular bacteria.